Welcome to the Tree of Life podcast. My name is Joel Litford, and I have the privilege of being here today with Professor Laura Bogar. She's a professor in the Department of Plant Biology. Laura, welcome.
Thank you, Joel.
So today I wanted to spend some time getting an idea of your academic journey, how you ended up here at UC Davis, and maybe talk a little bit about your research. I'm really interested to know more about mycorrhizae, and I think you're the perfect person to ask about that.
Yeah, that sounds great.
So can you give us a little information on what your academic journey was? What were you like as an undergrad? Where were you? Where did you go from there? And how did you end up at UC Davis?
It's a good question. It's been a few years. So I started out as an undergraduate student at Lewis and Clark College in Portland, Oregon. So it was a small liberal arts school, about 1,600 undergrads at the time. And the biology department, of course, was much smaller even than that. I was interested in going into something biology-oriented when I arrived. I thought I might want to be a field biologist in a park or a ranger or maybe like a high school teacher. But as I started taking classes and got to know the professors, they suggested I might want to try biology research as a way to engage more with the subject matter and explore other career options. So my first research experience actually was focused on spider evolution. So I didn't start out working on fungi. I learned some basic molecular biology and evolutionary reconstruction techniques with Professor Greta Binford there, and subsequently decided I wanted to focus more on sort of plant systems was what I thought. I was interested in plant ecology, and Professor Peter Kennedy was there working on, drumroll, mycorrhizae. So I was interested in the outdoors. I was leading mushroom identification hikes for the College Outdoors Club, but I wasn't an expert. I could tell which mushrooms were edible and which ones weren't, but not much more than that. Working with Peter, though, I learned how to combine plant seedlings and fungi in experimental settings and use those experiments to understand some of the mechanisms driving ecological patterns that we see, and starting to explain the things that really intrigued me about finding mushrooms in the woods. Why is this mushroom always under this tree and not under another tree? Why does it only show up in a certain time of year and not in the summer? So science and research in plant-fungal relationships started giving me the tools to dig into those kinds of questions.
So do you think that when you were an undergrad just starting, did you have kind of a natural curiosity of when you were outside walking around, seeing living things, not necessarily fungi, but whatever?
Absolutely. Yeah. I knew that I enjoyed spending time outdoors, and I knew that I always wanted to know why things were the way they were. And in a liberal arts context, it can be kind of hard to focus on which sets of why questions you really want to dig into. And that remains true for me. I have selected science as my path, but I'm really interested in why our social systems are set up the way they are and why political structures are the way they are. And that's not how I'm trying to spend my professional time. But relentless curiosity is a trait that I see across a lot of not only scientists, but all kinds of people in professional research roles. Yeah.
Yeah. So you mentioned systems, and I just kind of use that as a nice segue. So I think of you as a systems biologist in our department. And can you give us some context? What does it mean to be a systems biologist?
That is a great question, Joel, because it's something that I have discussed even with the other systems biologists in our department. If you sign up for a class in systems biology, which I may be helping to teach in the next few years, you will learn most likely about interactions between different players inside of a cell. So typically we use this phrase to talk about understanding genes and proteins and metabolites within a cell. And it separates itself from traditional molecular biology because instead of focusing on what happens when gene A and gene B are both expressed at the same time in a sort of pairwise way, the systems approach starts to account for all of the gene products that those are making, all of the proteins and metabolites and genes, and the ways that they can interact in unexpected ways. when we think about them as a whole system instead of a set of pairwise interactions. So, and I mean, the system can emerge from the pairwise interactions. Basically, I think of systems biology as being what happens when molecular biologists use the statistical tools that ecologists and computer scientists have been using to describe complexity for a while. Essentially, I'm trained as an ecologist, and I think that systems biology allows us to use mathematical tools to understand all kinds of complex interactions, whether we're talking about genes within a cell or organisms in an ecosystem.
That's really interesting. So when I think of systems biology, like, you know, in plant biology, we have a new major, systems and synthetic biology. So it seems like, based on your description, there is kind of that hopeful connection with synthetic biology as well, right? So maybe re-engineering some of those pathways and things that you discover as part of systems biology. Is that fair to say?
Absolutely. So systems biology, especially with the slightly narrower understanding of systems biology as being the ecology within a single cell, understanding emergent properties of these complex systems allows us to engineer what that cell is going to do much more effectively than if we try to pretend that each gene and each protein exists in isolation. If you start to study how genes and the proteins that they make are regulated and the many, many feedback loops that happen, it becomes really clear that we need to account for that complexity when we think about generating new functions in that system. You want to make sure that the product you're trying to make isn't going to accidentally repress its own production somehow or interfere with other important cell processes that are happening. So the systems approach allows you to hopefully model the cell with enough detail that you can actually predict what's going to happen and manipulate it intelligently.
That's really amazing. So then, just to connect that with your focal group of interest, fungi. So can you, for us, tell us a little bit about your study organisms, and really, I'm super interested to know about mycorrhizae more. When I teach mycorrhizae, I mean, I know some things. I know that it's a really ancient symbiosis. I know that mycorrhizae have lots of benefits to plants. My dad's a farmer, and so oftentimes farmers add mycorrhizae to try to, bolster the symbiosis. Can you tell us a little bit more about mycorrhizae?
Absolutely. So I am aware all the time as a new member of a plant biology department that I work both on plants and on fungi. And my training has been from a fungal perspective, and it's very exciting now to have colleagues who are focused on the plant side because the mycorrhiza is plant and fungus together. Nearly every plant that you see has mycorrhizas on its root system or mycorrhizal fungi. And they are an ancient symbiosis. It's been hypothesized reasonably, I think, that land plants themselves were able to move from the ocean into more terrestrial situations in part by partnering with these fungi, which allow the plants to functionally kind of extend their root systems. So this is a symbiosis where the fungus grows out into the soil and it gathers nutrients like nitrogen and phosphorus and even water that a plant might need, and it can bring those back to the roots. And in exchange, the plant, which, as we all know, is going to be really great at photosynthesis. So the plant takes carbon out of the atmosphere, turns that carbon into high energy molecules like sugars and lipids, and sends those to the fungi on its roots. So you have this reciprocal movement of resources that sometimes can be thought of as kind of an exchange that allows the plant to benefit by getting more sort of soil resources, and the fungus to benefit by getting the energy it needs to survive. So it's ancient. It's nearly ubiquitous. Almost every plant has it. And there's a lot of different kinds. I work specifically on mycorrhizas that involve mostly forest trees and these large-bodied fungi called ectomycorrhizas. Those are less common than the broad pattern I just described. Most plants have a different kind called arbuscular mycorrhizas. And there's some interesting differences that I could get into, but it's good to know that when you go out for a walk in the woods or in any landscape, you're walking across not only a bunch of root systems, but fungi attached to all of those root systems.
Now, I've watched some documentaries and read some papers and some popular science books that sometimes describe the mycorrhizal network almost like an internet connecting all of the plant individuals. How true is that? What is your take on using it as a sort of sending communication signals between individuals in an ecosystem?
It's a great question. That's been a focus of intense public and scientific interest over the last few years. I didn't really describe anything about what fungi are earlier, but it might be useful context that fungi not only make mushrooms sometimes, but they grow like threads in the soil. And so this sort of wood wide web hypothesis would say that these fungal threads would grow from one tree root system to another tree root system. And being little tiny threads, they might be kind of like wires, and you could imagine signals moving from tree to tree. We can produce these common mycorrhizal networks in greenhouse experiments. And if you do that, you do see relatively modest but non-zero amounts of resource being redistributed between different plants. It is possible for solutes, anything within the hypha. The hypha is the name for the fungal thread. It could move, or things could move on the outside of the hypha in one of these shared networks. The tricky part is that it's very difficult to know for sure that you have a single continuous thread between one tree and another tree in the forest. So that's a major research gap right now. We know that in theory, it's possible for plants to be connected in potentially functionally important ways. But when we go out to the woods, you can take a root from tree A and another root from tree B a few meters away, and you can sequence the fungi on those roots and discover that genetically those fungi are the same individual. But to know that those fungi are actually connected is a whole other set of experiments that are very tricky to do. So it's a hot topic in the field right now, and I would encourage any students who are interested in that sort of thing to both read things by Suzanne Simard and the groups that are really thinking about the wood wide web as a very reasonable model for the function of the forest, and also to read some of the work by Justine Karst and Jason Huxama and Melanie Jones. They're putting out a paper that should be out now or in the next couple weeks that really examines the limitations of the wood wide web as a model and the experiments we need to do to really understand how it works. So I think fungi probably are important. I mean, they are critically important soil organisms. They mediate the plant interaction with the soil in important ways, and they very likely do mediate plant interactions. But whether that is direct or indirect and what role direct connectivity plays remains an open question.
So can you tell us a little bit about some of the active research projects that are happening in your lab now?
Absolutely. I am still new in the department, so we are getting a number of things rolling, and it'll be interesting check back in a few years about which of these things have actually taken root and become the foundation of the lab. Right now, what I'm really interested in is trying to understand some of the mechanisms that allow plants and fungi to interact and what changing those mechanisms does to the function of a small community or tiny ecosystem. So to that end, I have projects going. The students who are working in the lab are working hard on developing methods to let us do these experiments. So it's possible to grow many of the fungi that I work on Petri dishes, on their own growth media. And we can also grow tree seedlings without fungi as long as we are delicate with them and give them some fertilizer. So if you walk out into the forest, you're never going to find one of these ectomycorrhizal fungi without a host plant, and you're also not going to find a host plant without a fungus. But in the growth chamber, we have more flexibility. So we're growing these two organisms independently, and I have a couple of students right now who are working to combine them in a very careful, sterile way, where we're going to study specifically how the plant and the fungus change the use of their genomes, so the expression of their genes through time as they associate in symbiosis. This has been examined in a handful of plant mycorrhizae interactions, but the fungi have evolved the ectomycorrhizal lifestyle something like 76 times independently. It's dozens and dozens of independent origins across really diverse fungi. So what that means is we might be looking at really diverse symbioses. There may not be an ectomycorrhizal symbiosis. I think we probably have diversity, and I'm interested in learning how variable the plant's response to fungi can be and how variable the fungal response to the plant is by doing experiments with a developmental time axis as well, so that I can understand how changes in gene expression proceed as the plant and the fungus interact. So that's a very microscale project.
Wow, that sounds amazing. It sounds really complicated because you're looking at things at such a big scale. I guess one question I would have is, do you find that there are, given that ectomycorrhizal symbioses have evolved multiple times, do you find that there are multiple ectomycorrhizae at the same time on a single individual? And do you think that there's interactions between those? Is it sort of like a mycobiome?
Absolutely.
Okay.
Yeah, it's a mycobiome, and there's a mycomicrobiome too. I have a rotating student right now who's excited about the fungal bacterial interactions that can make a huge difference in symbiotic outcomes. But yes, one tree in the forest, depending on the tree and the forest, but it will usually have dozens of fungal species on its root system at least. And those fungal species could represent hundreds of genetically distinct individual fungi. So fundamentally, a lot of my work is motivated by trying to understand why there are so many fungi coexisting on plant roots and in the soil ecosystem. It really seems as if, from a plant's perspective, if that plant could just have one best fungus, like the optimal symbiont, probably the plant would prefer that. But that's not what we see at all. We see great diversity. The fungi are competing actively for space on the roots and for soil resources. And so I'm really interested in building on the one-to-one experiments that we're doing, continuing with experiments where we put multiple fungi on the plant and see how the simultaneous presence of multiple directly or indirectly competing fungi changes the plant's response to those fungi and its management of that symbiosis. The plant doesn't have perfect control over the symbiosis. I'm also interested in where the limits of that are. So we're going to use gene expression in some of those careful experiments that I described, and also stable isotope enrichment to keep track of actually how much carbon that the plant fixes through photosynthesis goes to any one of the fungi on its root system.
Oh, amazing. Okay.
Yeah.
So, okay, here's your final question.
Okay.
It's the zillion-dollar question.
Mm-hmm.
Okay, so let's pretend like the chancellor comes to you and says, Professor Bogar, I'm ready to fund any project at any level that you need for, perpetuity. What is that project? What does it look like?
Wow.
The biggest idea.
the biggest idea in mycorrhizal symbiosis. Oh, I feel like anything I say is not going to be thinking big enough because the chancellor has very deep pockets.
There you go.
One thing that I'm trying to work towards that would be exciting, although if the chancellor were to come to me, I hope he'd send me an email so I could think about it a little bit longer. What I'm working towards right now would be the dream of really being able to understand in real time what fungi and plants are doing when they interact in the field. What I want to be able to do is pull a root from the forest and analyze it. Ideally, this will be some kind of nucleic acid sequencing. I think RNA sequencing is a good target for this, but maybe it's metabolites or proteins that are going to tell us the answer that I want. But I want to be able to analyze it in some kind of straightforward way. And from that, I want to know how much carbon is the plant putting into that fungus? How much nitrogen, phosphorus, water is the fungus bringing back to the plant? Is that symbiosis functioning well in terms of the sort of immune response of the plant? Is the plant accepting this fungal partner, or is it actively trying to defend against it? I would love to be able to go out to the field and really understand plant and fungal behavior, for lack of a better word, using some of these high throughput sequencing techniques that we have. That's ambitious, but doesn't feel quite impossible. If the chancellor has additional funds, I
Sure.
would love to get a better understanding of what controls plant fungal compatibility. So in addition to wanting to know what they're actually doing out there, I would love to know why some fungi grow only with certain plants. We see whole genera of fungi that can only grow with genus Pinus, and often you'll have individual species that can only grow with like a 2-needle pine. And the plants usually are not so specific, but I think if over the next few years we are able to better develop some transformation tools and engineering tools for these actomycorrhizal fungi, we could start to understand what are the sort of genetic tools the fungus needs to associate with a given plant, and what really are the trade-offs between remaining a generalist who can grow with many plants or specializing on a particular host. Fundamentally, a fungus, if it wanted just maximum access to plant resources, you would think it would want to associate with every kind of plant that it can find. And we don't see that happening very often. Usually the fungi are specialized to some extent on a particular host plant, and I'm really interested in understanding what mechanism drives that. How common is specialization really, and how is it enforced over evolutionary time?
That's amazing. I mean, it sounds like that would really get at the heart of characterizing symbioses just in general.
Yeah, I think a lot of people interested in symbiosis are really interested in this specialization question, and the mechanisms do vary by system, but it's really unclear right now for mycorrhizae what the trade-offs are and why the fungi bother to specialize. So I'd love to understand that.
Sounds fantastic. Well, thank you very much for coming on today. We look forward to hearing of your future discoveries. Please, we'll provide a website for Professor Bogar's lab. If you have any more questions, feel free to send her an email. Thanks for joining us, and we'll see you next time.
Thanks, Joel.